friends, welcome to Shri Ram coaching classes. Our today's topic is molecular basis of inheritance. In previous videos, we have learned about the experimental proof that DNA was a genetic material. This experiment comprises of three different set of experiments. The first was performed by Griffith in year 1928, and the second was performed by Avery, MacLeod and McCarthy, and the third was performed by Hershey and Chase in year 1952. I will just only give a gist about these experiments in this video tutorial. You can watch my complete video on each experiment separately. So let's start this experiment. So there are three experiments for saying that DNA is a genetic material. The first is by Griffith in year 1928. Okay, so let's start. So I am just giving a gist what was happening in the Griffith experiment. The first he take M for mouse plus S strain of bacteria Streptococcus pneumoniae. Okay, in this experiment, when the mouse was inserted with the S strain of Streptococcus pneumoniae, the mouse died. Okay, in the second case, when he take mouse and inserted it with R strain is for rough strain. It is non-virulent form of the Streptococcus pneumoniae, and this is virulent form for Streptococcus pneumoniae. As we know, this is non-virulent, so mouse will not die. The mouse is alive in second case. Now comes the third case. Mouse plus heat killed S strain. Now what will happen in this experiment, the third case step of this experiment is when we insert heat killed S strain of Streptococcus pneumoniae, the mouse will not die. It will be alive cause the bacteria is got killed in this case. Okay, so now in the fourth step of this experiment, mouse, we take these two strains, R strain and heat killed S strain plus heat killed S strain. So this is we took both this and this completely, so in a complete set. So what will happen? We know that R strain is non-virulent, so mouse will not die, and we know that heat killed S strain is not responsible for making any disease in this or disease in mouse as known as Streptococcus pneumoniae. So in this case, we know that mouse will not die. But when we took the first one and this one and this one together and inserted in a mouse actually died. This was very surprising experiment. As we know, this is non-virulent and this also become non-virulent. But the complete set what we got here is that the heat killed S strain bacteria cause transformation of R strain bacteria and cause it from non-virulent to the virulent form. This was transformation principle that was given by Griffith in year 1928. So here comes the second experiment of this transformation principle, that is Avery, MacLeod and McCarthy experiment in year 1944. So in year 1944, Avery, MacLeod and McCarthy worked to determine the chemical nature of transforming principle. So they purified this sample, this R strain plus heat killed H strain, and purified it to 99.98% of purity. So they just subtracted all the protein content from it and get it 99.98% pure form of this heat killed S strain and R strain. So to understand what is causing it non-virulent to be a virulent bacteria. Okay, so let's start. They purified it and started the experiment in three steps. The first is they take protein, this sample, and use protease. We know protease are enzyme which will digest all the protein in the content. Okay, so there will be no protein, so the sample will contain no protein. Then also we found that this sample can cause transformation. This is causing transformation. So this is not the factor or character which was using. the procedure of transformation, so making non-violent to violent, because we can see in this case there is a protein, and when we use protease, the protein get digested, so leftward portion still can cause transformation. So this protein is not a thing that is genetic material. So now comes the second case. We take RNA, they take RNA and use this sample and use RNAs. So in this case also transformation is going on, it is taking place at a very smooth rate. So this is also not a factor which was causing transformation. So now comes the third case. They take DNA and use DNAs. DNA is the same case. These are also what? Let's start. These are also the enzyme which will digest all the DNA content from the sample, and the left out portion will not have a DNA at all. So when they performed this third step, what they found was transformation was stopped. There was no sign of transformation. So this was clearly proved that only DNA is a genetic material, not protein, not RNA. Cause in the absence of protein also, there is transformation taking place. In this case, transformation is not taking place. In both these cases, transformation is taking place. And in the absence of RNA also, transformation is taking place. But in the absence of DNA, no transformation is taking place at all. Okay. So let's start the third experiment of transformation series, and that is Hershey and Chase experiment. Hershey and Chase performed this experiment on bacteriophages in year 1952. So the question that is arising is, what are bacteriophages? Bacteriophages are virus that specifically infect bacteria only. Okay, so I just should draw the structure of bacteriophage. This is bacteriophage, okay, and this is second bacteriophage taken in this experiment. Okay. So what Hershey and Chase did was, they take bacteriophage and radioactively labeled this one with sulfur 35. So let's take sulfur 35, radioactively labeled part. This is sulfur 35 in protein coat. This, okay, this is all the bacteriophages protein code. See why this experiment is going on with using bacteriophage, cause bacteriophages are only made up of two things. The one is protein content in their cell wall, and the second is genetic material, DNA, in their inside capsule. So this is their DNA. So they clearly, very clearly prove that only DNA. In the following manner, I will suggest you how they goes on the experiment. So what they did is, they take radioactively labeled sulfur 35 and inserted in their protein coat. Okay, and in the case of this one, second case, they take phosphorus 32 and radioactively labeled phosphorus 32 and inserted in their DNA. Okay, so let's start the experiment. The experiment proceed in three phases. The first one is infection. As I have earlier told you that bacteriophage will only attack specific bacteria. Let's take in this case E. coli. It will affect E. coli. Okay, so I have drawn E. coli, and for this case also E. coli. Let's start. Now this bacteriophage will come in this case and in this case. Bacteriophage will come and will infect our E. coli or bacteria. This E. coli is a bacteria, okay, will come and infect our E. coli. Okay, this is our infection phase. The E. coli get infected. Now comes our second step, that was blending. As the name suggested, blending. Okay, what happened in blending phase? Virus coat removed from the bacteria. So this is our bacteria E. coli, and also in this case E. coli. Now this bacteriophage, what happen is, when bacteriophage if infect the E. coli, it will transfer its genetic material here. It will transfer its genetic material here. Suppose this is bacteria's own genetic material, this is, and here comes the genetic material of bacteriophage. Here comes genetic material of bacteriophage. This is genetic material of bacteriophage. Okay, And this is genetic material of E. coli itself. So now in a second phase, blending, these bacteriophages will be just removed from the bacteria, not separated from the sample, but just removed from the bacteria. So they get removed from the upper bacteria. They get removed. Okay. This is their genetic material, and now here is the bacteria's own genetic material. Now comes the third and most important step, which will determine that our final conclusion. That is centrifugation. So now what will happen? So in centrifugation, we know that we have just inserted phosphorus 32 in the DNA of the bacteriophage and sulfur 35 in the protein coat of bacteriophage. Okay, so if we get protein in our result, then we will say that protein is a genetic material because it passed from bacteriophage to E. coli. Okay, and if we get phosphorus 32 in our E. coli cell, then we will say that DNA is a genetic material because it passed from the bacteriophage to E. coli. Now let's look at the result. So results were like, we found no radioactive sulfur 35 in the E. coli and detected the supernatant radioactive 35 in the supernatant form of the virus. Okay, the parallel experiment what we found is that the radioactive phosphorus 32 is detected in the bacterial cell and no radioactivity shown is supernatant. So we unequivocally proved that from this, with the help of this experiment, that DNA passed from the DNA passed from the bacterial cell to our E. coli, and hence only DNA is our genetic material, no protein at all. Thank you.